Alright, my name is Devin Harmon, and this is a presentation on excitation-contraction coupling. So, what is excitation-contraction coupling? Basically, it's a multi-step process, ultimately resulting in a skeletal muscle contraction. So, when you decide to bend your arm by contracting your bicep, as you can see here, that's excitation-contraction coupling, which sends that signal. Specifically, it's the activation of peripheral nerves, which are innervating skeletal muscle to get them to contract. A signal is sent from your brain to your skeletal muscles to cause that contraction. And the process itself begins with a neural impulse in the form of an action potential in the motor neuron. But an action potential doesn't just spontaneously generate. To initiate excitation-contraction coupling, a series of steps must occur to cause this. So now we get into the initiation of an action potential. So an action potential is created through the process known as neural transmission, and it begins with the activation of a signal from the brain or spinal cord. This signal initiates the opening of voltage-gated ion channels in a motor neuron. However, not all signals will cause this. The signal must reach a threshold of negative 55 millivolts. As you can see here, the signal must reach this threshold in order for an action potential to be generated and the channels to open. If this threshold isn't met, then there will be no action potential generated and no voltage-gated ion channels will open. So action potentials follow what's called the all or nothing principle, meaning that if this threshold is met, then a full action potential will generate. All action potentials are the same size. So as long as the threshold is met, then it'll generate to its maximum size. All action potentials have the same size. It's not like a shorter, like a smaller voltage will result in a smaller action potential. As long as it meets the negative 55 millivolt threshold, all action potentials are going to have the same size. But if it doesn't meet the threshold, then no action potential will be generated at all. So that's why it's called the all or nothing principle. Regardless of the strength of the stimulus or the size of the neuron, all action potentials will have the same size. Although it should be noted that increasing the strength of the stimulus will increase the frequency of an action potential. So when this threshold is met and an action potential is generated, this results in the opening of voltage-gated sodium channels and a sharp influx of sodium into the cell. This will cause depolarization in the neuron as the membrane potential becomes increasingly positive. So because sodium is a positive ion, that having a rapid influx into the cell will depolarize the neuron and make it more positive. After some time, between 0.5 and 1 milliseconds, the potassium channels will begin to open as well, causing potassium to efflux out of the cell and kind of reverse this depolarization process. Since potassium is also positive, this will create a flow of positive ions out of the cell and reverse this depolarization, which will cause, as you can see here, this drop from the action potential. It's known as the repolarization period. And also around the time that the potassium channels open, the sodium channels begin to close. So that's what initiates this repolarization phase as the voltage in the neuron decreases. During this repolarization period, the membrane will rebound towards its resting potential of negative 70 millivolts, but what actually happens is the potassium channels stay open for a little too long, and it will actually go beneath that. And it will take a bit for it to return to its negative 70 millivolt resting potential. And that period from which it goes from beneath the 70 millivolts back to its resting state is known as the refractory period. And this lasts about 2.5 milliseconds after the stimulus is introduced, or 1.5 to 2 milliseconds after the opening of potassium channels. When the membrane returns to its resting potential, the neuron can prepare to fire again, and another action potential can be generated. So that's how an action potential is generated, but what does this mean for the muscle? So the generated action potential travels down the axon of the motor neuron until it arrives at the presynaptic nerve terminal. And this is the end of the neuron and the portion that communicates with the muscle at the neuromuscular junction. So this down here, this is the muscle fiber, this is the motor neuron. This area where the motor neuron and the muscle fiber communicate with each other, this is the neuromuscular junction. So when the action potential reaches the terminal, it triggers the activation of these voltage-gated calcium channels. Calcium will influx into the cell and interact with these blue acetylcholine packets that exist in the terminal. Acetylcholine is the neurotransmitter which is involved in excitation-contraction coupling. It's synthesized in the motor neuron cell body and then transported down to the terminal where it is stored in these vesicles. So acetylcholine is released from the motor neuron through a process known as calcium-stimulated acetylcholine exocytosis. So this process occurs in three main stages: docking, priming, and fusion. During the docking phase, the acetylcholine vesicle is moved near the presynaptic membrane, and the snare proteins begin to interact with each other. The snare complex is a group of proteins which function to help fuse vesicles to a target membrane. The main ones that play here are VAMP, also known as synaptobrevin, syntaxin, and SNAP25. VAMP/synaptobrevin is located on the synaptic vesicle, while the other two, syntaxin and SNAP25, are located on the presynaptic membrane. These three proteins work together to permit fusion and facilitate the release of acetylcholine into the muscle membrane. Following docking is the priming stage, in which the snare complex assembles and prepares for fusion. Another protein called complexin comes into play and binds to this complex in order to prevent premature fusion. Fusion isn't meant to occur until calcium binds to synaptotagmin, so complexin comes in to regulate that. During the fusion stage, a new protein called synaptotagmin comes into play, and this is basically the calcium sensor. Calcium will bind to synaptotagmin when its levels in the cell become high enough. Synaptotagmin interacts with the snare complex in order to induce synaptic vesicle fusion, meaning that acetylcholine is released into the synaptic cleft to bind to the muscle. The synaptic cleft is the space between the presynaptic nerve terminal and the postsynaptic muscle membrane. After acetylcholine is released in the synaptic cleft and begins to move towards the muscle fiber, SNAP proteins and NSF, which stands for N-ethylmaldehyde sensitive factor, bind the snare complex using ATP so its components can be recycled. Acetylcholine binds to the postsynaptic muscle membrane via acetylcholine receptors, which are located in a specialized region called the motor end plate. In order for excitation-contraction coupling to continue and for a muscle contraction to eventually occur, we need to generate what's called an end plate potential. So, in order for an end plate potential to be generated, about 100 vesicles need to release within the same millisecond. This will activate surrounding voltage-gated sodium channels. So the resting state of the motor end plate is about negative 80 millivolts. So you can see here. And in order for an end plate potential to be generated, that needs to go up and reach the threshold of negative 55 millivolts. And only when this threshold is met will the voltage-gated sodium channels be able to open and excitation-contraction coupling will continue. When this happens, it depolarizes the sarcolemma of the muscle fiber and an action potential is generated. And this generated action potential is what will eventually cause the muscle to contract. Acetylcholine esterase is an important enzyme in this process, which works by degrading acetylcholine after it triggers an end plate potential in order to prevent it from overstimulating muscles. It's a very active enzyme which hydrolyzes the bond between acetyl and choline in an acetylcholine molecule in order to inhibit its activity. The action potential generated from an end plate potential will propagate from the neuromuscular junction outwards in all directions and will be conducted into the muscle fiber by the transverse tubule system. It takes about 50 milliseconds to fully depolarize the longest of skeletal muscle fibers, but on average it will only take about 6 to 7 milliseconds. So the sarcoplasmic reticulum basically serves as a depot for calcium in the muscle. The sarcoplasmic reticulum feet are what anchor and connect the sarcoplasmic reticulum to the transverse tubule system. The SR feet contain the ryanodine receptor and dihydropyridine receptor, which are two key proteins in this process. The dihydropyridine receptor acts as an action potential sensor. So you can see the action potential being conducted across the sarcolemma here, being sensed by the dihydropyridine receptor, and the ryanodine receptor acts as a calcium release channel. So the action potential which comes into the sarcoplasmic reticulum will be sensed by the dihydropyridine receptor, which will in turn open the ryanodine receptor calcium channels. Calcium ions are then released into the myofilament region of the sarcolemma, as you can see here. So now we get into the myofilament interactions which actually cause the force production. The released calcium ions will bind to troponin, which is an important regulatory protein for muscle contraction. Troponin releases its inhibition of the actin filament, thereby enabling filament interaction between actin and myosin. This allows for cross-bridge cycling and thus force generation. These interactions between actin and myosin will continue to occur as long as there are neural impulses at the neuromuscular junction and high calcium concentrations in myofilament regions. So if I go back up to this slide, you can see here the calcium in the myofilament regions. This cross-bridge cycling and force production will continue to occur as long as calcium is in this region, but when calcium is no longer in this region, we'll see what happens. So here's where ATPase comes into play. ATPase is an important enzyme which acts to pump calcium back into the sarcoplasmic reticulum when neural activation stops. So all the calcium that was in the myofilament region is going to be pumped out of that region back into the sarcoplasmic reticulum by ATPase as soon as neural activation stops. This obviously results in a decrease of calcium in the myofilament regions, and thereby inhibition of the actin-myosin interactions will resume. So these two filaments can no longer interact with each other, so cross-bridge cycling and thus force production can continue to occur, and force relaxation will ensue. As a side note, muscles with a high SR calcium pumping ability will be able to relax sooner since they can get the calcium out of the myofilament region and back into the SR sooner, meaning they can relax more quickly. Alright, so there you have it. That's basically excitation-contraction coupling. In summary, excitation-contraction coupling is the process by which a signal is converted from the brain into an actual muscle contraction. A neural signal from the brain or a spinal cord will generate a peripheral nerve action potential. This causes the influx of calcium into the presynaptic nerve terminal, which will trigger the release of acetylcholine. Acetylcholine will move across the synaptic cleft and bind to acetylcholine receptors on the muscle fiber in the motor end plate region. This allows an end plate potential to be generated, which results in the depolarization of a sarcolemma and the generation of an action potential. This action potential is conducted into the muscle fiber by the T-tubule system. The dihydropyridine and ryanodine receptors in the sarcoplasmic reticulum feet will sense this action potential and in turn open calcium channels, which will result in the release of calcium into myofilament regions of the sarcolemma. The released calcium will bind to troponin, which will then permit myosin-actin interactions, which result in skeletal muscle contraction. When neural activation stops, ATPase comes into play and pumps calcium out of the myofilament region back into the sarcoplasmic reticulum, and actin-myosin interaction is blocked again. Forced relaxation will then occur, and the muscle will stop contracting and begin to relax. So that's it. That's excitation-contraction coupling. Thank you for watching.